Hello, I'm Robert, and back in the day when I was a child, then I would have been very excited by this idea that is going to classify much of Mars like the Moon, and humans can go there and we just not have any problems. But I have a bit more background since then. I've become interested in planetary protection in the last few years, and I always had a deep interest in science. Science also has great commercial value potentially if they would discover a microbe on Mars that works differently from Earth microbes, not based on DNA, not based on RNA, then this could be the basis of billions of dollars industry, even if you just look at it from a commercially. We have $1 billion industry for enzymes from extremophiles that originally we wouldn't have if it weren't for studying extremophiles. And what we could find on Mars could be far more exotic than that. And we can't invent life. We could find life on Mars that's just beyond anything we can imagine. And even it's just a microbe, and inside it's not like any Earth microbe, even not remotely resembling. And we can't even make an Earth microbe from scratch. You can't just you can make a virus, you can make a DNA strand, or you can make an RNA strand. You can't make a complete living cell. We just can't do that. So you know what it's made up of, you know what all the chemicals are. If you try mixing them together, they'll just react together. You get a mush and you won't get a cell. So we can't invent new forms of life. And what we could find on Mars could be absolutely unique. So we have to be careful. And so first of all, so this proposal is to recognize Mars as being like the Moon. And I'm going to explain why it was run by a planetary geologist. For some reason, they didn't choose astrobiologists to do this study, and they're using out of date. They used a 2014 report without taking account of a review in 2015. Even as that report was being published, NASA and ESA found problems with it. So I'll talk about that, and they don't even mention those. So it's very, very patchy and incomplete, the research they did. I'm assuming they're being honest and straightforward, and they only knew about the 2014 report and didn't know about the 2015 review. And there's been so much since then also that they just didn't know about in this report. At least don't mention. Anyway, I think part of this may be because they want to return a sample from Mars to Earth. And NASA want to do it by 2030, I think. And if you go and look at the NASA documents, they have loads and loads of papers about how to design the sample return vehicle, which doesn't even exist except as paper studies at the moment. But they haven't yet started the legal process. And the legal process is too late now to finish by 2030. If they wanted a sample returned by 2030, they should have started the legal process by 2020. By 2010, I mean. It's just far too late. And there's no sign they're even aware that they need to do this. This thing, perhaps they expect it to be like Apollo 11, where they didn't even do a proper law. They just had a kind of informal internal document they published on the day that Apollo 11 was launched. And it didn't go through any proper legal process. And that wouldn't be permitted today. We have learned so much more since then about how the environment can be vulnerable to things. And people are very keen on the precautionary principle and just being very taking precautions even when you don't know that for sure that what you're going to do can be harmful. And so in the backwards direction, from Mars to Earth, we just don't know that if there is any native Martian life, if it could harm human beings or the environment of Earth. And as an example, there are likely to be biofilms on Mars if there's native Martian life. One disease of biofilms on Earth is Legionnaire's disease. And Legionnaire's disease can also infect the human lung, which is a kind of biofilm as far as this disease is concerned. It is not adapted to the human lungs, although because of the way it is now being spread in human habitats and through human lungs, and then back to our habitats again. It is evolving to be able to make better use of that as well. Well, Martian life, Martian biofilm could evolve to make better use of human lungs to reproduce. So that again doesn't rule out Martian life. And then in addition to that, if we had Martian life that was let's just keep on that thing. If we had Martian life that was adapted to if we had Martian life that was based on different biology, then our antibiotics would be useless against it, most likely. Even Earth life manages to evolve defenses to antibiotics by developing alternatives to the cell processes that the antibiotics target. If it's an exotic form of life, all the cell processes are likely to be already alternatives to the cell processes targeted by our antibiotics. So you wouldn't be able to stop any Martian diseases of biofilms from invading your lungs by using antibiotics. And likely wouldn't be any medicine you could use at all to do anything to it. And there's no reason, the two possibilities, Earth life could be completely, Martian life could be completely mystified by Earth life. Or it could be that Martian life doesn't even notice, Earth life doesn't even notice, it's the Earth life that's mystified. It doesn't even notice that Martian life is life. And that, or it could be that they're rather equal, but those are two possibilities. And then the Martian life could then just punch away at your lungs. And your lungs don't even notice that this is life, it's just trauma. And it wouldn't mount any attacks, any defense. It is possible, and it can't be ruled out, that if astronauts were to land on Mars today, that they'd be dying even a day or two after they first come in contact with the Martian life. These planetary protection provisions are not just paperwork. There is a reason for them. And then it could be perfectly harmless to human beings, and then whatever you return from Mars to Earth, it might be harmful to the sea, for instance, to little sea creatures, and harmless to humans. And it could take part of our biosphere and our bodies, and then get back to Earth, and it gets into the sea, and then it affects the fish, it could affect the soil. And then one way to think about how it could be harmful, then imagine if Earth microbes and Mars microbes were just about equal in capabilities. And we ended up with Mars microbes return to Earth, and they adapt and they spread. And it does actually get as warm on Mars as it gets on Earth. It can get 20 degrees C, and even a bit more in the tropical regions on Mars. And then there could be geothermal vents below the surface. There could easily be heat-tolerant microbes on Mars that, just like that on Earth, that can withstand very high temperatures. It can probably withstand the oxygen, because there is oxygen in the Martian atmosphere. The surface is very oxygenated with the perchlorates and the hydrogen peroxide. And this new research suggests that there'll be levels of oxygen in the brines on Mars as well. So Martian life could easily be capable of living in coexisting with Earth life. Now imagine that in your stomach, for instance, maybe there could be the possibility of sulfate caves on Mars, and whatever life would live in those would be acidophiles that would be able to cope with high levels of acid, just like it is on Earth. So again, you could have acidophiles on Mars, and they could live in our stomachs maybe. Now imagine your stomach, and you have equal amounts of Martian and Earth microbes there. Then those Martian microbes, maybe they have perchlorates inside them, where Earth life has chlorides. It's quite likely, because there's loads and loads of perchlorates on Mars, and there's very little by way of chlorides. It's quite reasonable that Martian life might take it up and use it in some way, and this is not very good for humans. So, and then you get, for instance, accidental poisonings, like the microbes, the blue-greens, the cyanobacteria blue-green algae that's spread across the Great Lakes and these are poisonous to dogs and to cows. And they damage their, they kill them. Anyway, they can kill them. They can damage their internal organs. And then so there's no guarantee. And then now if you imagine that our Earth biosphere is transformed, you return microbes from Mars, it's a clash of biospheres, and you have equal numbers of Earth and Mars microbes everywhere. The Mars microbes are not based on DNA. They've got something different inside them, and they produce completely different signaling chemicals. They may have perchlorates instead of chlorides. There may be all sorts of differences. They maybe can't be eaten by Earth life. Maybe Earth life just can't make anything of it, or maybe Earth life produces no defences to it. Then are the Earth biospheres then, are the Earth ecosystems still going to function? And the very worst case. So remember that we are completely protesting against this. So I help people get scared of such things. So what I'm describing is potentially very scary, but it is not going to happen. NASA do not realise that how strongly protected Earth is from legal processes. And what I'm saying now, if there's any validity to it, then as we talk about the NASA sample return, then all of this will be played out and discussed in publicly and fully. And it's only when we completely work through all this. So already with sample return missions, then the previous studies by the National Research Council in the States and European Space Foundation, they have both found it's very important to protect Earth against Mars microbes. I'm not sure that they have quite they quite appreciate quite how bad it could be in the worst case. And I've got an article that I'm preparing that I hope eventually to get published. I was encouraged by an astrobiologist friend to give it a go. And so I think there's a possibility that, and this is based on, for instance, there's a physicist, Claudius Gross. He talked about how if he was looking at sending life to transiently habitable, that's transiently habitable for a few million years, distant star systems, and looking at the clash of biospheres there, and he came to the conclusion that it is possible that after a clash of biospheres, there would be the microbes of both sides would likely adapt to each other quite quickly because they evolve quickly. But higher life forms, and they maybe wouldn't be able to adapt in time, and there might be almost no multicellular life left after a clash of two biospheres. That's the worst case in the return direction. Now, so we definitely have to go through a legal process to check that this is not going to happen. The easiest way is to search for life in situ on Mars. And I don't know why NASA are doing sample return, because all the astrobiology papers I've seen say that's not the way to do it. They say that in these very harsh conditions here, we're not likely to find the life there very easily. If it's very easy to find the life, then maybe you can return it as sample return. But if it's even moderately difficult, and unless you've got a way to find it in situ, so they say you should find it in situ first. 3 papers I've got that I found on the subject. I didn't find a single paper by an astrobiologist saying we've got to do the sample return first. And we've got wonderful, very tiny instruments now we could send to do this. So it's all very bizarre. I don't really understand why they're so focused on the sample return. But they're not going to be able to do it unless they sterilise it. So we don't need to worry about that. And so my main effort is in the forwards direction. And I have a whole article about that we can go into. I don't want to spend too much time on this. I spent a quarter of an hour on that. Didn't mean to spend so long. But it's relevant to what's going on, what I'm going to talk about next. So in the forwards direction, legislation is very weak. And sadly, it's all the Outer Space Treaty. And so it would be possible for them to achieve what they're doing in that direction, and that's what I'm so concerned about. And in the forward direction, so they came up with the idea of doing And they used this report from 2014 by Rommel et al., which proposed using maps to divide Mars into special regions. And they suggest basically using these maps and then classifying these as category 2. In other words, and category 2 means that there's, go up to category 2, as the absence of niches where places where terrestrial microorganisms could proliferate and to a very low likelihood of transfer to those places. None of the astrobiologists are saying this. No astrobiologist is saying nowadays that there's no possibility of habitats. The ones who think that maybe there aren't habitats, the ones who think that the potential habitats may be uninhabited. But I haven't found any astrobiological recent papers saying that the Mars surface is that there's no possibility of habitats. We just don't know yet. And in 2015, even over those regions, the 2015 review says maps can only represent incomplete knowledge, and it's based on these streaky things, also called the RSLs. And during the day, then during the year, then they start off very small and short. They grow and they broaden and they darken and then they fade away. Now, this is very difficult to understand. It's not like what happens with dust. I'll just search for dust. It doesn't seem to get in there. Yeah, dust cascades. So there's a new idea that perhaps these are caused by dust cascades. This is my my paper preprint that I was talking about, and so it's possible it could be related to dust cascades. But the study finds some features that are consistent with dust cascades because they terminate at slopes that match the stopping angle for granular flows of creationless dust. It seems a bit of a coincidence if it wasn't dust cascades. But there's an awful lot that isn't explained there. Oh, what happened to that? Oh yes, here we go. So this is what you get with dust. These dark streaks that's a pure dust avalanche, an avalanche slide. So if it's just really dust cascades, just an avalanche slide, and that's all it was, that's what you expect. And you'd expect it to stay dark for decades. But instead, these fade away. This is another one of them. See how it just fades away at the end of the year? That is not what you'd expect from a dust cascade. So this, they're also remarkably narrow and consistent in width. And again, you don't expect that from a dust cascade. No, this is more like what you'd expect from dust. And it develops seasonally. There's no reason for dust to avalanche seasonally. So they start when it warms up, when the surface above gets to about 0 degrees centigrade, which would be below the surface, then the binds are a little bit, some very cold binds maybe, and it's hard to know quite what's causing it. But there are hydrated salts associated with it, which suggest some form of liquid water. Part of the problem is that the spacecraft that photographs these is in a sun-synchronous orbit that comes close to Mars only during the summer, in the mid-afternoon, early afternoon. And if there's liquid flowing there, it's in the early morning, and we just can't get close-up views of it. It's in a big elliptical orbit. It's always, when it comes close to that side, close to the Mars, is in the early afternoon there. And those are our best photographs of the RSLs. So it's very hard to know what happens in the morning, which is the most likely time for flowing liquid. So the leading hypothesis of these is still that they correlate in some way with the seasonal, with seasonal flows of brines. And there's this is quoting from my own paper, preprint. and because I'm quoting from literature that's surveying that. And so if you just read the news from the NASA press releases, they don't mention these points, or barely touch on them if they do at all. But if you go into a literature survey, you find a whole dialogue, and there's some arguing one way and some arguing the other way. And it certainly is not resolved, and the dust cascades was just one that for some reason got publicised quite widely. And other people arguing against it and finding problems with it, and they both have problems for how you supply them from the top, where you get the dust from, and where you get the brines from to keep flowing down. Both the problems, getting enough dust to keep flowing down, or getting enough brines to keep flowing down. So it's extremely difficult still to know what's going on there. So, anyway, so that's and then the thing is that they found that there are, when Curiosity landed, then these were found after the Curiosity landing, and the same thing, dark, narrow streaks. And these could be RSLs near where Curiosity landed. This means there could be habitats there for that Earth life could survive in. And this is why the 2015 review said that we shouldn't use maps. It said only use as in preliminary, what they said exactly about maps, that they can only represent the current and incomplete state of knowledge for a specific time, knowledge that will certainly be subject to change or be updated as new information is obtained. And this is an example of new information for Curiosity. And if there is any life in these streaks, then it's just below the surface, probably a centimetre or two, maybe even 10 centimetres below or something. And this would not be something you could see from orbit. Even in deserts on Earth, which are far more habitable than these conditions, you can't expect to see the life from orbit. It's under, a lot of it is below the surface of the desert, or under the crust of rocks. And even if the tiny patches on the rocks, they wouldn't be kind of, you get to a third of a metre scale from orbit, you wouldn't see much by way of life on that kind of resolution. You wouldn't see a patch of lichen, for instance. So there's vigorous debate in Nature and in Astrobiology Journal again, and it started in 2013, and again this is not mentioned in this report. But both sides in the debate are agreed that if we bring humans to Mars, they will introduce Earth microbes there, and this is and this is just not even and that. Of course, we'll bring Earth microbes there, but then that we'll irreversibly contaminate it. And that if there are any habitats there, which are well possible, then their idea is that really the debate between these two parties in this debate, the debate is between one side who think that that human beings are going to go to Mars in the next decade or two, and that once humans get there, we'll have lost our opportunity to study Mars in its pristine state. And so they say we must send rovers there, and don't worry if they're a bit dirty. And even if they're going to, even if we know they're going to introduce microbes to Mars, it'll be nothing like the microbes that will come on a human spaceship and give us an opportunity to study Mars in a relatively pristine state before the humans get there. The other side in this argument say that we do have plenty of time. Humans are not going to get there as quickly as that, and that we can clean our rovers properly and study Mars properly. I would say my personal view is neither of those. I think that we need to convince the Mars colonization enthusiasts that they should do things differently and focus on the moon. So I'm saying both of these, don't do either of those. Do it properly on Mars. The life on Mars matters. And these Mars colonization enthusiasts, they're not experts on astrobiology. They don't know a thing about astrobiology, for the most part. Tell them how astrobiology works, and tell them that we need to know what's there before we land humans there, and tell them that there is a risk to the astronauts themselves. Because it's very hard for them to believe this, because we don't encounter other biochemistries on Earth. We've got nothing to prepare ourselves for what could happen when we go to Mars. We have nothing even remotely like this in any of Earth's history. And these people claim that Earth and Mars have exchanged life, but if you look at it closely, it's not at all clear we have. If you look at the whole process of an asteroid hitting Earth, sending material off to Mars, then the shock of how it exchange it has to sudden acceleration, that kills most microbes. Then it goes flying through the atmosphere. It has to go faster than the fireballs come in order to get out of the atmosphere at escape velocity. So it'd just be a fireball going through the atmosphere, the entire surface of it covered in plasma, and would go in little cracks. And then when it gets to the Mars surface, then the possible habitats there, you've got little rock inside. If there's anything left inside, we won't get the earliest things maybe get there in decades. Most would take hundreds of thousands of years, and in very dry conditions there. And maybe there is little brines there, but is the life going to get into it? And if it gets into it, bear in mind the Chicxulub impact was into a shallow tropical ocean. Is the life that happens to have survived all that and got to Mars, is it going to be pre-adapted to Mars and be able to live in the conditions there? It's very, very different from landing there with a human mission with whatever microbes we come with us. It could be anything. And we have extremophiles in our spaceship handling facilities and on our bodies. They're just mixed up all over the place. Life that has adapted to live in very hot temperatures can also retain those capabilities in normal conditions. It's a very different situation. And as for the trek coming back from Mars to Earth, then these habitats, just dust and ice and salt. It's not easy for those habitats, life in those habitats, to get back to Earth. And then we have no idea what the Martian capabilities is. Maybe the shock would kill it all, even though it would kill the shock of impact and sending material off to Earth from Mars would kill nearly all photosynthetic life on Earth. There are only some very resistant microbes which would survive it. And the Martian meteorites we have from Mars, they come from a couple of meters below the surface. You can tell that by the ionizing radiation patterns on them. They can't be near the surface, but you'd see it on one side and have a side. It would have an asymmetry to it. So on Mars, they would bury deep below the surface, and they come from the high southern uplands, where the air is very thin, because that's where it's easy to be thrown off into orbit. And it's also easy to understand. If you've got an impact, then the bit that flies up into space, it generally comes from below the surface, not the surface layers. It's generally below the surface layer that gets up into space. And that's what we've got here. And that again is a place where there's not very likely to be much life on present-day Mars. And as for in the past, we don't know if early Earth life or early Mars life had the capabilities to go back and forth. The most likely time for it to happen is very early solar system, but we don't know if it happened then either. And astrobiologists, when they design their instruments for Mars, they design them in order to be able to cope with any form of astrobiology, and they do not assume that it's in any way related to Earth life. So, anyway, so this debate is just not mentioned in this report, and lots of ways that there could be life on Mars. And this was Nilton Reno, and that's Nilton Reno there in the background there, and he was a principal. He's one of the best experts on liquid water on Mars. He's in charge of the weather station on Mars, on Curiosity, and he was principal investigator, I think, for the Phoenix lander, where they observed these droplets as salt and ice was thrown up onto the legs of the Phoenix lander. And then they formed these droplets, which seemed to form very quickly. And he's tried to simulate them in his experiment, and he found, this is just quite recent, just a year or two ago, that after a few tens of minutes we've got salt in contact with ice, which could happen over large areas of Mars, but not in equatorial regions. Then within a few tens of minutes, you get liquid water forming, liquid salty water, which could potentially be habitable to a microbe. And as he says, it's just a droplet of water. It's like a swimming pool to a microbe. So, and then as we go, and then I say he was in charge of the vegetation on Mars, and they've had another remarkable discovery, and that's in where Curiosity is. And they discovered there that that if it travels over rocky areas, and then it travels over a sand dune, then the humidity just does a nosedive when it goes over the sand dunes. And the explanation of that is that the perchlorates in the sand dunes, which are taking up water from the night. And you may know that I'm in a higher latitude in Scotland, and here we have, like, it can be dry in a sunny day in the daytime, your clothes are drying. If you leave them overnight, then you can get a nighttime frost, and you can get dew and frost on the grass. And because at night, as the air gets cooler, then the humidity increases. It's the same amount of water vapour. There's no new water vapour, but just colder air contains less water, and so it becomes more humid to touch, and eventually water will come out of it. Well, on the Mars surface, then you've got these perchlorates which actually take up salt water, just like if you've got salt in a very damp place, then it'll get kind of sticky and it won't pour out very easily. Well, this is like that. The perchlorates, they take up water very avidly, and the below 15 degrees centimeters depth, they now think that the Martian sand dunes, there's permanent liquid there, liquid brines. But it's very cold, too cold for Earth life to accept. It's possible that below -20, it's possible that Earth life does reproduce with a timescale of maybe centuries to thousands of years. But it's almost impossible to do the lab experiments to check that. And so what they say on Mars, they generally take this view that with planetary protection, that I described that there, they generally, have I put that in there? They generally take the view, I'll say it anyway. Planetary protection is basically keeping it free of Earth life only for own purposes, and not for future generations thousands of years from now. So they can't have it as being okay for planetary protection if the conditions are so cold that life would take millennia to colonize it. So Earth life might possibly be able to survive below -20 degrees C, and not just survive, but actually colonize it very, very slowly over thousands of years. But that's just ignored. I don't think we should ignore that, especially when going to Europa and Enceladus. My personal view is that we should have completely sterile rovers and not have the possibility of Earth life being introduced even very slowly. But that is how it is at present. And so the perchlorates, they take up the water at night and they dry out in the daytime warmth. And in the top 5 centimeters, now I can just bring up the actual picture in the paper. There we are. You see, this is where they found the liquid at 0 centimeters and liquid at 5 centimeters below. So it's dark blue, the liquid at 5 centimeters below. And this is the year, so summer, spring, winter, autumn. And that's sunrise and that's sunset. And that's when the sun is shining. And as you can see, these areas they can have liquid right at the surface at night, right at 0 degrees centimeters. And then as it dries out in the daytime, and by then, soon after sunrise, then it's too dry, but it's still liquid at 5 degrees centimeter in the winter and spring especially. And then it becomes too dry then as well. Now, unfortunately, or fortunately, however you think about it, for Earth microbes, then when it gets warm enough for Earth microbes to sort of do their thing and metabolize sort of above that -20 degrees centigrade, then it's too dry. In fact, it's zero water activity, absolutely no possibility. We need 0.6 for Earth microbes. On the face of that would say, oh, that's completely safe, there's no possibility of Earth microbes. And here you can see in the sunset, for some reason, it's only in winter, but in summer it's a much broader period in which you've got it. And so we'll go and look at that. So we're back here, and that's their schematic diagram they did probably for the press release. Nighttime water goes in, daytime it dries out. However, we don't know what the capabilities of Martian life is. I mean, for all we know, Martian life is able to survive down to minus 40 degrees centigrade, remember, and even lower. Maybe it's the Martian life there just permanently, just doing its thing at minus 40 degrees centigrade. There's a proposal by Dr. Schulz-Markuk that we could have perchlorates and hydrogen peroxide inside that life. He thinks that life would, if it was living at those low temperatures, that it would just kind of disintegrate when raised to normal temperatures, and that could explain some of the Viking results. But if I remember right. Anyway, it's suddenly disintegrating at high temperatures. It just sort of reacts with itself. But that's not absolutely essential. There's also perchlorates are a chemotropic agent, and that means that it enables metabolic processes to happen more quickly. That could even work with earth microbes, and it's not so damaging it. And cold conditions is more enabling of life. And it's possible that earth microbes on Mars, the experiments just don't seem to be done. This was a question that was raised in a 2015 review. Could earth microbes in the Martian conditions, taking up the perchlorates, have faster metabolic processes than they do on Earth, and be able to do more metabolism at lower temperatures? And they recommended that we should do experiments on that. I don't think these experiments have been done. I can't seem to find them. If anybody watching this knows, then do let me know if there have been experiments in using perchlorates mixed with earth microbes and seeing if they can have faster metabolism at low temperatures. And if there's some microbes that could take them up and use it, even if some can't. Anyway, whether earth microbes can do it, then it's entirely possible that Martian microbes can. Now, Nilton Renault has suggested, and then it could be returned to Earth, so there could easily be life. It's possible, and this, if it was below 15 degrees centigrade especially, the successor to Curiosity, as far as I know, the sample return mission is not going to be drilling into the sand. It's just going to be drilling little cores out of rocks. And the rocks, so it would miss all this. If there's life there, it's not going to find it. It might find some spores in the dust. I don't know if they're going to bring back dust, but their priority, they're thinking like geologists, and they're looking for rocks. And it seems their only aim is to return rocks, and not the dust. I don't think they're going to return any of the dust. Astrobiologists want to drill deep, but the geologists don't seem to be so bothered about that. And then, do the low nighttime temperatures really make the Curiosity brine sterile? That is a big question. So Nilton Renault, as I told you about, he's the expert. He's an expert. Suggests that earth microbes might be able to exploit the liquid brines through biofilms. And this was also mentioned in that 2015 review that is being ignored by this report. And they say cells in biofilms embedded in the matrix externally produce substances produced by the cells, polysaccharides, proteins, lipids, and DNA outside of the cells. And they can adjust the environmental parameters to make the entire biofilm more habitable. And they say many examples of this on small scale, even micro-scale communities on Earth with biofilms only a few cells thick. And they can propagate in these biofilms even with adverse and extreme surrounding conditions. So Nilton Renault now is suggesting this. And he's saying that while the new study interprets Curiosity's results to show that microorganisms of Earth would not be able to survive and replicate, Renault sees the findings as inconclusive. He points to biofilms that can make their own microenvironment. So it's entirely possible that Earth microbes, I'm not sure whether he's talking about Earth microbes or about Mars microbes there, it's just someone talking about what he said, but the Earth microbes could probably do the same trick, but there could be Martian life there already. It has formed a biofilm. So remember, as I said, if I go to this is just theoretically, not detected. They haven't detected these drops. Theoretically, even the surface should be liquid at 0 centimeters. Now, if there was a biofilm there, it wouldn't release the water, it would trap it. And even at 0 centimeters, it would seem that in principle, a biofilm could trap the water through to the daytime. And so you could even have like photosynthetic life covered with a millimeter thickness of dust, which would help protect it from ultraviolet light. You only need partial shade. We have life on Earth that is tolerant of certain levels of ultraviolet light, and so Martian life would be at least as tolerant as Earth microbes, and we have cyanobacteria that can tolerate quite high levels of ultraviolet light. And we have lichens that could survive on Mars conditions in semi-shade. So there's no real reason why this liquid couldn't be held in biofilms, or even a photosynthetic life right up in the surface, within a millimeter of the surface, and retain liquid during the day. And it's only the water vapor. All we know from the water vapor is that that it's driest in terms of not evaporating like perchlorate would do. But the biofilm wouldn't. The outside of the biofilm would be dry. Inside it's doing its thing. So I don't think that we have proven that there's no Martian life there, and I certainly not no Martian life. I don't think they've proven that it can't be contaminated with Earth life. Indeed, I mean, I think there's a possibility that Curiosity could have contaminated. You can't say it hasn't, but hopefully, if it has, it's not going to be. Gilbert Levin, he thinks he's concerned that maybe we've already contaminated Mars to the extent that the Viking landing was the first and only study of pristine Mars. Hopefully, it's not as bad as that. And if there is, I think personally that the top priority thing we should do is send a mission, but it should be 100% sterile. I'm just going to talk in a bit about how it is possible to have a 100% sterile mission to Mars. I think we should be focusing on 100% sterile missions, actually. And I think one of our top priorities should be to send a life detection mission 100% sterile in order to find out how well our planetary protection is working. And a good place to go would go to the Phoenix lander, which got crushed by the advancing dry ice, and its solar panels broken off, and so the little crushed up bits of spacecraft in a place where we know there are these little salty swimming pools for bacteria, and it's possible that it's been contaminated with Earth life to some extent already. Now, I mean, the hopeful thing would be that it's very, very. There's a good chance that the life just wasn't adapted. That what we've sent there already, although we do have life that could survive on Mars, and that could have got on our spacecraft, there's a good chance that whatever actually has got into this hasn't been adapted. That the ultraviolet light and the harsh conditions there have been sufficient to keep Mars sterile of Earth life. But I think we should study and see what's happened. And so I just explained that. Let's go to that at this point. Right. I'm just wondering where to go now. As you understand, I'm not planning this. I think it's better I would take. It's best. I think it works best just talking spontaneously. So, yes, what's the problem with bringing Earth life to Mars? I'll talk about that in a minute. But let's just look at this. Why do we do it? 33 seconds in.
Microbes to Mars in order to sort of test whether Mars could be a habitat.
Yeah, some people
Mars
think
could be a habitat, whether
Mars,
whether it could be a habitat for Earth organisms. That's something we might do eventually. If the international community decides it's the right thing to do, we can certainly do it. It's just that as we go about the process of exploring Mars, we don't want to screw up the things we want to do first by doing things that then we can't take back afterwards. You can't do a do-over on releasing organisms in the Mars environment. Once they're there, they will be there. So we have to do all of our search for life activities. We have to look for the Mars organisms without the background, without the noise of having released Earth organisms into the Mars environment. This is why we are very careful when we clean robotic spacecraft, because we really want to understand what's there at Mars and not see the stuff we brought with us by accident.
So that's why I've got to be so careful. And so yes, so what was I talking about? I can't remember what was I talking about just then. The perchlorates, and well, I'll just keep going, and then I'm sure I remember. So then then there's I thought I would tell a little short story at this point that about what might happen if this is something I think is a simplest possibility. This could be a real future. It is consistent with our best current understanding of Mars. So this is called Prestige or Dishonor, First Footsteps on Mars. That's my little story. And it's just told through some newspaper reports. I pretend I'm a reporter reporting the first astronauts on Mars. So the first astronauts land on Mars, plant flags on the soil. And I didn't want to do it for some particular nation. Didn't want it to be the US flag or something. But this is a flag of the Earth. And who knows, it's far too expensive for one country to probably do it. So maybe we will have a flag like that. Maybe, probably would have all the other ones as well. But it'd be nice if we had one of those. And then that'd be very exciting. I can imagine a child being very excited by that. But then the Mars astronauts, they find life. Wow. And you can imagine how excited everyone would be, except for perhaps those amongst the knowledgeable astrobiologists, like the ones with that big debate. They would probably be somewhat dreading what happens next. Big disappointment. The Mars life was from Earth. Because they brought there in a dust storm. Now, you get people saying that humans would not bring much life, much microbes to Mars. They seem to think that we could just travel around on Mars and not bring any life there. But this is not what happens. And they have this, Robert Zubin talks about this expedition, an SUV that went across the Arctic or something, and they did these, and they did searching to see if they'd left any, looking for microbes. But what they don't explain very clearly is that they went and searched only once. They didn't go back again the second time round and do a second SUV run. They just spent the day there, went out and looked very far, a few meters away from their campsite, or from where they were, and they found that there was no life there. They didn't look again later on to see if it got moved in winds or something. And they went on and did another one and another one. But this is what happens. These are some people who are actually studying microbes in the dry valleys in Antarctica. And so it's very important to keep the Earth contamination. They want to know what the native microbes are like. So they don't want to contaminate it with their own microbes. So they have these little corral systems with their tents and a 50 square meter site. And with a 50 square meter site, they reckon that after a 10-day camp, typically they have 60 billion microbes, an average of 100,000 per cubic centimetre, which with the very sparse populations there can be between 0.1 and 10% of the natural population. And remember, the populations of Mars would like to be even more sparse than we have in these dry valleys. So there is a significant risk of overloading even the region where the camp is, where they land, if there's life there. But then there's the Martian dust, and the Martian dust is very, very fine and just spreads like that's an artist's impression, not a photograph, but it's like that. Fine, it's as fine as cigarette smoke in extremely thin atmosphere there. Fine, fine dust. And yet enough to protect it. It's iron oxide in it, protects it from UV. And as you say, so then big disappointment. And it was so sad, just an earth microbe that stuck along for the ride as a hijacker. And it's just explaining how easy that is for that to happen. And then that native Mars life found, the ups and downs, very ups and downs in this story. We've gone and found native life now. And it contains RNA and ribozymes, but no DNA, and no ribosomes or proteins. So ribozymes would be capable of, they're made of little bit fragments of RNA stuck together, and these would be capable of maybe catalyzing exact reproduction of RNA and a gene sequence. And it could do it without any DNA, just these single-strand RNA reproducing without any DNA, and without all the complexity of it unzipping and the for producing messenger RNA which goes off the ribosomes, which turns into proteins. There wouldn't be any proteins, there wouldn't be any ribosomes, there wouldn't be any DNA. A much simpler form of life. And then so they find this. And they would be able to find out. They would see some cells swimming around, and yet these cells, they find there's no DNA and no ribosomes, and they find there is RNA there. So if you imagine they had a reasonably equipped lab there that they brought with them, they might have found this life, and maybe they found it by sort of drilling a little bit below the bit where they found the life before, or in a different part of the RSL. They found a bit we hadn't got to yet, very, very sterile instruments. Maybe they sent a sterile robot to find it for them. But this life is no match for Earth life, because there was once an idea that we would have a shadow biosphere, and that's where this photograph comes from. At one point that was thought to possibly be an RNA world life. Those things which I forget what they're called, from a new form of life, maybe a great find of the century, that was once thought to be RNA world life on Earth. So if it's possible on Earth, it's certainly possible on Mars. And maybe it is there still, but it's gone extinct on Earth, and whatever made it extinct on Earth would likely make it extinct on Mars if we introduce Earth microbes there. So that's a significant risk. And you can argue either way, but it's certainly possible, and it could be a very early form of life. And the earliest form of life, then it could why should, this is one Paul Davies saying, why should proteins be universally necessary? It could be that Martian life has no proteins, and it could be RNA form world life. And he thinks that the LH84001, it might be fossil organisms from Martian RNA world, and maybe that's still there. And these, if you go back and look at it, you took modifiable cells. The cells would start off probably not even being really cells, but just little vesicles, abiotic vesicles, and then eventually, then they might become somewhat biological and produce their own cell walls. But they're still just easily. They don't have defenses. They just take up. They have little vesicles. They have little vesicles inside them. Sorry, and then yes, the replicons, small virus-like replicons, and they would be populated abiogenic lipid vesicles. The tiny little you can get things that are like cells that form through non-biological processes, and maybe that's what formed here, and maybe that's what we'll find. We might even find things called apoietic cells, which are even more primitive, where they don't yet reproduce exactly, or not at all. They just reproduce, they sort of grow and split, but there's no exact replication at all. And that's also possible. And if we find anything like that on Mars, it would surely be extremely vulnerable to Earth life. Especially if you imagine even the earliest one, before there's even any exact replication. It just seemed like a kind of organic soup to Earth life. Just eat it up and it's gone. And I think that it's a possibility that we lose it as easily as that. If we feel there could be something very interesting. And people often talk about, well, what if Mars has uninhabited habitats? Well, and perhaps life has gone there extinct, and then it just arose again in the last million years. Well, on Earth, we can't replicate an entire planet of habitats with Earth, with life arising again, even in a million years. So this is still something that of extraordinary interest. If we find something that's almost life, or something that's very, very primitive form of life, maybe evolved for 10 million years, 100 million years, even just a few million years, and whatever it got to, And even if it didn't get to life, what does it get to? So, and that in a way, in some ways, it's just as interesting. Indeed, if Mars has no life, then this maybe gives us an idea of what many Mars-like planets in our galaxy are like. And it'd be extremely interesting to know how far it has evolved chemically, and whether it has these autopoietic cells that people theorize about, whether it does anything that is a kind of predecessor of life. And this would be extremely interesting. Mars is the only Earth-like planet we have, apart from Earth itself, in our solar system. And if we introduce Earth life to Mars, and if originally it had no life, and it just had abiotic processes, this is our last chance, short of being able to travel for light years, of actually studying a planet like Earth without life on it that has never been modified by life. So that is extraordinary interest as well. Just we don't have anything like it, nothing in the solar system that is like Mars. And we can't, as Cassie Conley said, we can't do a do-over. Whatever we do to Mars, and then these dust storms, they spread throughout Mars. Suppose that we send humans to Mars and they crash. The first spaceship there crashes. This is well possible, because it's extremely difficult landing. This then likely stops human space flights for maybe a decade or so, just like happened with the space shuttle. And during that decade, you have these all the spores from the crashed spacecraft spread over hundreds of square kilometers of the Mars surface, spreading around in the global dust storms. And the dust storms, they travel so fast that I can't remember, I think it's a week or two, that a dust grain can go all the way around the Mars equator and be back again where it started. It's entirely possible during a dust storm, and dust storms start in one place on Mars and they spread over the entire planet, from which it is obvious that dust grains can travel fast enough to get anywhere on Mars. So if you imagine several global dust storms later, then there would be spores from Earth life almost anywhere on Mars. There's a possibility of when you're considering these billions. Remember, it was 160 billion spores, and then imagine a crash on, was it 160 billion? No, 60 billion, sorry. 60 billion microbes left in the soil. And there would be many more than that if you've got a crash of a human spacecraft with all the supplies and the food and the soil maybe for agriculture and the bodies of the people, sadly. So it is possible that the first human landing is, and so anyway, so our Mars life is found. This is not such a bad scenario as that. Humans did land successfully, they found life, but they weren't able to get it. It's doomed to extinction on Mars, and then they maybe aren't able to replicate it, because we have lots of microbes on Earth that we're just not yet even, I think it's 40%. I'm not sure if I put it in here. I think I'll put it in this article now before I publish it. I forgot about mentioning that. But there's a study of the gut microbiome from 2019. So that's a very recent one. And estimate, I think, about 40% of the microbes in the human gut can't yet be cultivated in the laboratory. And an awful lot of them, I think a similar percentage, is that it's not been DNA sequenced. You just get little fragments of DNA. So it's very well possible if we find some novel, completely exotic form of life on Mars, and yet it does perfectly fine in our guts. These microbes are very happy there, but we can't get them to reproduce in the laboratory. So similarly, there could be life that is very happy on the Mars surface with all its other little pals doing its thing. And maybe there's even some pioneers that if you get them in the right conditions on Mars, there must be pioneers that are able to spread around and start and form pioneers in very harsh life conditions, as we consider it, on Mars. That doesn't mean that we can get this to work in a laboratory. even trying to reproduce the Mars conditions, then consider these are just astronauts landing on Mars. They could keep it alive all the way back to Earth. It's entirely possible that it's all dead before it gets back to Earth. So, and that then could be the last we ever see of Martian life. And this is not impossible. This can happen. This can actually happen. This could be a future that we read in the news in our future, and maybe a decade or two from now. And astronauts, of course, regret making Mars life extinct. And it's just so sad. And can we do anything to protect at least some of it? So far there's been no success in getting to replicate it in the laboratory. Because of dust storms, it's spread all over, and we've had global dust storms since then. So this is the sort of thing that we are risking. And this is a rather sad story. It's very hard to find any treatment to this in science fiction. Most people just optimistically, like in Star Trek, they don't even think about microbes. They just beam down to the surface, and then they go around, and there's no even problem of clashing biospheres. They haven't even thought of the idea, except where it's needed for a plot point, in which you have some very specialist microbe that just so happens, and yet they're happily drinking each other's food and everything as well. It could be different biochemistry. They have a kind of plot, I don't want to go into it, but they have a kind of idea behind that of the ancient hominids, which are supposed to have engineered all the planets in the solar system, in the galaxy, so they produce exactly humanoids like us. So you can sort of make it plausible they all have the same life, because these ancient, very wise hominids who somehow knew how to cause life to go all the way forward and end up exactly like we are on many different... That's not plausible for our solar system, even for our solar system, for Earth and Mars, if it's independent. So anyway, and this was back before they realised quite how inhospitable Venus was, and it's the only story I know of this form, but they found this wonderful form of life. And then it's such a sad story, sad, sweet story. It's actually mobile. It's not impossible that Mars has mobile sponges and little worms. Now that we know it's the possibility of oxygen in the brines there, a new discovery last autumn for Vladimir Semenkovich, saying his name right. And so anyway, we could find little worms there even, and they could go extinct. Or some kind of animal life form. Maybe even things with little legs, who knows? I mean, that's possible. There's oxygen levels right in the polar regions are quite high. They could potentially go almost all the way up to little creatures that scramble around in the rather oxygen poor floors of the sea. Anyway, so it would have to be adapted to very cold conditions on Mars, or able to somehow, some way of warm water meeting cold water. Anyway, so they find this life form, and then they burn their waste, and then leave the explorers on Mars. Then they leave their waste food and some chemicals, and he finds phosphorus and stuff there, and it's this life form, not non-thinking. It's not a thinking life form. It's just blind instinct. It feasts on it, and it's able to eat all sorts of strange things. But then the bacteria and viruses, which on an older planet had evolved into 1,000 deadly strains. Though only a very few could survive in this heat and this atmosphere, they were sufficient. As the carpet crawled back to the lake, it carried contagion to all of its world. Even as the morning star set course for a distant home, Venus was dying. The films and photographs and specimens that Hutchins was carrying in triumph were more precious even than he knew. They were the only record that would ever exist of life's third attempt to gain a foothold in the solar system. Beneath the clouds of Venus, the story of creation was ended. We don't want that to happen on Mars. How sad that would be. And then you have this long debate about why did it happen and how did it happen. And I've got an analogy of the Lascaux cave, about how, keep an eye on the time, how Mars today is about like Lascaux caves, where whatever it is there in its pristine form after a billion years of life or inorganic chemistry. And then in Lascaux caves, it was just our breath, and unintentionally now microbes, just by visiting the caves, and then attempts to make it better actually made it worse. And it's so I've got that sad story. And if we can't, and then in a recent conference, climatologists said we can restore the original environmental conditions of the Lascaux cave, but the microbiologists say it's not possible to restore the microbial conditions as they were before the year 2000. So if we can't even do that with the Lascaux caves, what chance do we have of restoring Martian habitats to the conditions as they would be before Earth life was introduced? We certainly do risk, and there may be numerous treasures there, precious treasures like the Lascaux cave paintings, that would be completely destroyed by Earth life if we do this wrong. So what should we do? So I'm not saying that we just have to give up on human space exploration. Not at all. As I say, I'm a great enthusiast for humans in space. I think that we need humans and robots together. Humans are much more expensive, but if we can crack this, if we can have a sustainable, self-sustaining system. So humans are very expensive if you have to keep supplying them with food, like we have to do with the ISS. And I think we should have long ago already been studying self-sustaining systems, which the Roscosmos, the Russians, they have studied this and they've found ways of getting it work on the ground. We just haven't done it in space. They've had astronauts growing all their food from only 30 square meters, just about all their food. And then you eat the food, you produce carbon dioxide, and you produce human waste, you burn your excrement, and that then produces the carbon dioxide, you burn the plant waste. You don't really need to compost it, because the plants turn it all back into organic matter again. And like on Earth, there isn't really a great value in composting if you've got plenty of energy. So you can just burn it, and then life grows and takes it up again. And then if you produce all your own food, you automatically produce all the carbon dioxide it needs for the next cycle around, for the next generation of food crops. And if you import food, you're having an excess of carbon dioxide. So carbon dioxide you're not short of carbon dioxide in a human habitat, even if you're growing plants there. And it's not true that the Martian carbon dioxide in the atmosphere, that's of no value to human beings. We have too much carbon dioxide. Only tiny amount of it is needed anyway. 0.04% of our atmosphere. And the idea that you're going to Mars and you want some of that 0.04% of carbon dioxide to come from the Mars surface, it is useful for fuel, but you have far more fuel needed to get back from Mars to Earth than you need from the Moon to Earth. So anyway, so that's what I'm saying. We need to go to the Moon. And then, and Chris Hadfield put it like this. He said, I think ultimately we'll be living on the Moon for a generation before we get to Mars. So I think that the children who are following Chris Hadfield, and if it fits with everything that I know, then I think that the children who are born when the first astronauts start going back to the Moon will be the generation that the first astronauts that go to Mars orbit. I think they're far too optimistic, and that's why I say we've got loads and loads of time. They're not going to be sending humans to Mars before the 2040s. He says, I think ultimately we'll be living on the Moon for a generation before we get to Mars. If the world and the Moon were threatened, and the only way to preserve our species was to launch from Earth, we could go to Mars with yesterday's technology, we would probably kill just about everyone on the way. So think about Apollo 13. If we had anything like that on a spacecraft that was leaving Earth for Mars, then we don't have spacecraft at the moment with sufficient rockets to zoom back to Earth because you ran into a problem. Even if it was the very moment that you left Earth orbit, it's already too late. The only way to get back is via Mars. And so if you have chemical explosion, you have some problems with your life support, you have a fire. Fire is very dangerous on board a spacecraft. You have an explosion like I did with Apollo 13. You just have some problems with your food goes mouldy. or something, there's some chemical release that damages your food supplies or something. The ISS has had frequent problems with its life support. It's not so bad now, but in the early stages. And if we're starting off with a completely new spaceship like Elon Musk's Starshot, then its life support is untested. It's like the first time of the ISS. You can't test them on the Earth, not properly, not in weightless conditions. We just shouldn't set off unless it's, as you say, that the world and the moon were threatened. There's no way that's going to happen. And there's absolutely no way that the Mars can be a second home for human beings. No way. I mean, just look at it. It's very useful to have a backup in space, like sort of seeds. Seeds could be passively cooled on the moon, where it gets very, very cold. Cools liquid nitrogen permanently in the dark craters of the moon. A brilliant place for storing seeds for centuries. And that's a backup. And that's useful on Earth. Just an extra backup like we have on the Svalbard. That is worth doing in space. But it isn't worth thinking of space as being like a second home. I mean, you couldn't make Earth like Mars, even if you tried. You'd have to fire something like Vesta or Ceres at Earth to try and get it to like Mars, as uninhabitable as Mars. Or maybe, a very big asteroid. The Comet Swift-Tuttle could just about do it, but only for a few years, for a year or so. And it just doesn't make sense to go to Mars because you're worried about a big asteroid. Luckily, we know all the big asteroids, none of them can hit us for thousands of years. And if you want to protect Earth from a big asteroid, then send up lots of spacecraft. Indeed, the first thing is to detect them, and we won't detect the small ones for far, far less, even for less cost, I would imagine, than Elon Musk's. I think about the same cost as Elon Musk sending his Tesla motor towards Mars. Not actually to it, luckily. Then we could send up, I think, half a dozen or a dozen small satellites that would infrared spacecraft that would pretty much complete our survey. If we just done that as an experiment, why not send up a lot of infrared telescopes instead of my car? We know it might fail, but let's just give it a go in case. Then we could have that up there now, and within about less than a decade, then these small, using aperture, using synthetic tracking, then they would find nearly all of the very small, even the very small ones down to 20, certainly an awful lot of the ones down to 140, and a fair number down to 20 meters. And that's doing far more to protect Earth than trying to set up a colony on Mars. Elon Musk could do that tomorrow if he wanted to. Any billionaire could, take a year or two to maybe get them all made and then sent up in orbit. And that's far more useful. Protect Earth and then head off into space secure with a safe Earth behind. And the idea of making an entire new Earth-Mars atmosphere, that's way, way beyond anything we can do now. Look at how difficult it is to reduce the carbon dioxide in the Earth atmosphere by a fraction of a percent. It's 0.04%. And they're talking about creating an entire Earth thickness and pressure atmosphere on Mars. We're just nowhere near that. Absolutely nowhere near that. And their ideas, they just don't work. They take thousands of years, and they take 100,000 years to have oxygen there, and then Mars has half the sunlight of Earth. And that means half the heating relative to absolute zero plus a little bit of heat, internal heat. So it's far, far colder than Earth. And an Earth atmosphere on Mars is not going to be an atmosphere where you have trees. So you have to have artificial greenhouse gases there to try and keep it warm, or large planet-sized mirrors. It is not just a do-over of whatever they call it. I can't remember this American term for it. but is it Jova or something? Anyway, it's not just like an Earth Mark II. It's the Mars. So, as I say, we should go to the moon. It's as if you and I were in Paris, paddling around in the Seine, in little canoes saying, we've got pouts, we've got paddles, let's go to Australia. Australia? We can barely cross the English Channel. We're sort of in that boat in space exploration right now. A journey to Mars is conceivable, but still a lot further away than most people think. We need to go to the moon. And the moon, now I think one of the reasons why people are so, they feel that the Mars is, we've got to go there, is because it looks Earth-like. It looks quite a bit like Earth. They don't realise how very, very thin the atmosphere is, how that if you had scuba gear on the moon, I think many people would think you just go around on Mars with scuba gear and you'd be able to breathe it. No. It's so thin that your lungs would boil. You wouldn't be able to take in any oxygen, even if you have in your lungs already. You would only have the oxygen in your blood to survive on. You would soon faint. Then, and it'd be a good thing you did faint, because your eyes would be swelling, and the boiling, and your mouth boiling, and your body would swell up, and it would swell up to several times its size. Not several times its size, quite a bit, not several times its size. I can't remember the size. It's a significant percentage larger than it is, because of the swelling, because of the lack of atmosphere outside. And Mars' surface is not a place that you can survive without a full pressure spacesuit, which typically costs, you're to $1 million spacesuit, and even if they reduced it hugely in cost, it's so extremely complex. It's like a supercar in its complexity. You're talking about a couple $100,000 for, and then we're going to look in a minute how dusty these things would get. So apart from the atmosphere, which you can forget about, so then let's replace the moon by an Earth atmosphere, because the atmosphere is, actually the vacuum on the moon is a great advantage. You can use it for vacuum deposition and make solar cells, such as much better solar cells than we can make in our best factories on Earth, because it's such a hard vacuum. It's actually, and the reason it's not covered is not red. Red looks so attractive, but that's just rusty iron. And the iron on Mars is not rusty, so it's much more useful than the Mars rusty red stuff. This dark grey stuff is far more useful than rusty red stuff. It doesn't have the perchlorates in it. And then, so just add this, and you see it looks just like an Earth landscape. It's just as Mars-like if you change the sky. In fact, it's more Earth-like than Mars, because it's got these bright colours which you don't have on Mars. And if you could just have a kind of artificial reality kind of blue sky painted in as you walk around on the surface of Mars, of the moon. It looked quite a bit like Earth, if you wanted to look like Earth. So, and you see vacuum is a benefit, twice the amount of sunlight for solar power, lunar poles, the regions with solar power almost year round, 24/7, and there's nowhere like that on Mars. Not only that, I've mid-sentence. The CO2, see, I'm just drafting this and talking about this as a draft. CO2 is not much use, I explained. Pressure level is nowhere use. And then it's the dust. So this is Gene Cernan covered in dust on the lunar surface because the fender of his rover broke. And you see how it's completely covered in dust. Remember, this is going to be $1 million, maybe we could get down to a couple $100,000 spacesuit. So if you're an Mars colonist, you're going to be going through loads of spacesuits because they keep getting dusty. And you're going to have at least replaced most of your spacesuit every week or two. This is only after a couple of days. And now look at the dust. From the lunar rover. I don't know if there's any sound in this. I'll switch it off if there is. No, there isn't. So now just look as it comes close. See the dust pouring out as it drives around. See the dust spreading out behind. They call them rooster tails. And there's another bit, and then the other bits further on. See if I can find a nice good bit. I'll just leave it running while I talk. You see all the dust. So in the Mars surface, remember the picture of the dust storms? Then the Mars dust storms, then that dust isn't just going to on the moon because it's a complete vacuum. The dust does a little parabola and then hits the ground. There's nothing for it to hover in. So if you look very closely, each individual dust storm, you can't actually see individual dust grains, of course, but each individual dust grain, even though it seems like it's a great swerve of dust coming up, see? Great swerves of dust coming up. But each individual dust grain in that swerve is just doing a parabola. And that's all the dust, I think. And it's just doing a parabola as they drive along. But on Mars, you saw how the dust just hangs in the air. Then those great swerves of dust, they're just going to hang in the air, maybe for hours. And you're just going to be surrounded in dust wherever you drive. I don't think we'll really know until we have fast drivers on Mars. Mars is so slow that they can't just throw up dust like this. But it should be at least as dusty as the Moon. And the Mars dust storms, they block out the sunlight completely for weeks on end. And that's going to be bad for your solar panels. And so, as I say in my preface to Case for Mars First, you may have said I could say we should ignore the Moon, head straight for Mars. But actually, we're finding out that the Moon is far, far more interesting than you'd expect. It's resource-rich and fascinating. Many new discoveries and mysteries to solve. Potential for explosive metals and volatiles. Far more promising for tourist hotels and human research. I mean, tourists aren't going to go all the way to Mars, except very, very devoted tourists who are willing to have a holiday lasting a couple of years or something. Maybe retired people, like equivalent of cruises. But we could have weekends to the Moon. Certainly a week away, but it's a couple of days travel, but that could be speeded up. A weekend, not quite a weekend. You could have a week away to the Moon. You leave, if you have a holiday, then you could go to the Moon and come back again as a tourist. Once we've got faster spacecraft, we could maybe go away for the weekend. And it also, I don't actually mention there, but there's a possibility for astrobiology on the Moon. And that's at the lunar poles, where there's the permanent ice. Let's bring this up, have a picture to look at while I'm talking. So this is one of the most likely places. They're showing it. This might be the lunar poles or close to it. They're showing the Earth very close to the horizon. But it might just be, yes, because remember the Earth is always permanently in the same place as seen from the Moon. So the Earth being close to the horizon means it's likely either it's near one of the poles or it's near one of the terminators. But I would imagine it's near one of the poles. And so, don't necessarily mean to do that. So this is a possible lunar habitat, ESA's idea for a lunar village. And so this could be one of the poles of eternal light. I'm not sure the Earth is quite low enough. Maybe it is. Depends how magnified it is. Yeah, it's quite magnified, isn't it? Anyway, so that's, if we look there, the lunar poles, and they have these peaks where there's almost internal sunlight. The reason is because the Moon, maybe just by coincidence, but it's almost parallel to the Sun's axis. And it's it's tilted quite a bit relative to Earth's. So it's slightly, very, very slight, very little difference between summer and winter. And it has these peaks of almost eternal sunlight. Just a few days in winter when it's dark. And that could be maybe fixed by having something else beyond whatever's obscuring it, sending sunlight sending power back to where you are. So you could have solar power 100% all the time nearly. And you could have big solar towers, again, above it, which can clear away any nearby obstacles that there might be. And its temperature is almost constant, and I think it's minus 20 or something, which is very good, because actually the real problem for human habitat is you want to cool it. You couldn't get a more ideal habitat if it's in space for humans than at the lunar poles. There's nothing even resembling it on Mars. This is the place to go, the first place to go if you want to try a new colony. And admittedly, Mars might be better if you want to try 1 million people, but as far as, say, 100,000, then I would think the Moon's the better place. And they don't have any exports. Nobody has come up with any really sensible export from Mars. Deuterium doesn't work. I go through that in my book. I explain why it doesn't work. It's just not concentrated enough on Mars. You'd have to concentrate it even more, and then return it to Earth. You're just saving a few steps in what on Earth is a massive multi-story hundreds of tons, thousands of tons, I'm not sure, very big, complicated facility which you can never reproduce on Mars. No, there's no point in deuterium. And it's just slightly increased amount. And there's possibility of platinum on the moon, and there's very easy exports from the moon to Earth. Far easier than Mars. And there's a possibility of the Hoyt tether, which there'd be a tether, a rotating tether about the moon, rotating around Earth, Mars, the moon, and around Earth. And the moon is higher in the gravitational well. It's high up. Like you have to go upwards to reach the moon, as it were. And it's got very little gravity itself. If you could sort of throw material, it sort of would, there's a down towards Earth. It's going to gain energy as it's going down, falling down to Earth's potential well. And Hoyt has this very ingenious system of tethers, where one which would come if stationary relative to the lunar surface. So that maybe would be a landing place for it. And you put your materials onto it, and then it's just stationary just for a little while. You have to it'd be quite, maybe you have something that jumps up onto it, a little spacecraft that leaps up onto it, and then off it goes, and then it spirals around, and then it sends the materials off to the Earth in the distance. It sends off that, and it reworks it out, and it's not terribly long. It can be made with existing materials. It's nothing like the space tether. And then on the Earth you have the receiving one who receives it, and the receiving material from the moon boosts the Earth tether up into a higher orbit. And then it also sends material from Earth back to moon. And if you send enough material from the moon to the Earth, more than you return the other way, you actually generate power that way. You would even siphon off power at the Earth end, or at least use it to keep the system running, because it's downhill in the Earth's potential well. And he worked out, I think it says if you had exports from the moon every week for a year, then that's enough mass budget to make it worthwhile to build this tether system. It's not very heavy. And that would be almost free exports from the moon down to Earth. It doesn't seem to be much talked about, but it's a brilliant idea, it seems to me. It's far, far more interesting than the space elevator, which is talked about so much. And at the Earth end, then this thing can lift a hypersonic spacecraft from the Earth's atmosphere. Hypersonic jet that doubles as a spacecraft. It's just picked up by the tether, and it's powered by the material from the moon to Earth, and then from Earth it lifts things up. So you only need to go to hypersonic speeds, and you don't need to go to orbit. I think this is quite a likely system that we may have in the near future. Anyway, there are not so far from these peaks of eternal light, which this just possibly might be. I'm not sure if it is. There's certainly a possibility. Then there are these craters which is almost in turn totally dark, and where you get this is where you have it's totally dark in the crater, just a little bit of reflected sunlight from the surrounding mountains, and it's so cold, it's as cold as liquid nitrogen, and there's ice believed to be there. Well, that ice, if it's thick enough and the meteorites falling into it, then it can preserve organics from Mars, preserved, and from Earth. The Chicxulub meteorite impact on Earth, it should certainly have sent bits of ammonites and little bits of microbes and diatoms from the time of the dinosaurs, which would still have organics in it. On the moon, there may well be organics from the dinosaur era on the moon, pretty much preserved, unchanged since then. And there may be organics from Mars, which has got there, and it's got to the moon since the most recent meteorite to leave Mars to the Earth. It left, I think, 700,000 years ago. If there's any life here, anything on it, it's been sterilized long ago. But some meteorites from the Mars could get to Earth in a decade or two, a few decades, just like it does the other way from Earth to Mars. Especially a big meteorite impact on Mars. I'd have to look that up and find out what the actual figures are. But we could have organics from Mars that hasn't actually had much ionizing radiation actually on the journey either. And we could also, very early, we could have in the early solar system something that Venus reached its current state only a few 100 million years ago, and that before that it was not dissimilar to the Earth. If that's true, then asteroids hitting Venus could have sent life all the way to Earth, and to the Moon, I mean, not to Earth. It could have sent life or whatever was on Venus before then. So the Venus atmosphere at the moment is far too thick. But the Venus may have been like Earth, so Earth is covered with lots of limestone and chalk and various carbonates. And according to one theory, then Venus could have been like that too. And all the carbon dioxide atmosphere came as a result of a massive overturning of the entire surface of Venus. So we have these kind of superfluid things like the Deccan traps back at the time of the dinosaurs, but much, much bigger, covering the entire Venus surface. The entire surface, like the Deccan traps, are when large amounts of basalt came out to the surface and large amounts of carbon dioxide. But this is the idea that the entire surface of Venus sort of got trapped because it had no continental drift, according to this theory, and the heat just built up and up and up, and then it kind of overturned, and that an awful lot of carbon dioxide. Anyway, I don't want to go into that. I talk about that in my Touch Mars question mark book. And so anyway, there is this idea, for whatever reason, you can read out and find out more about it, but before then the Venus could perhaps have had life, and then that life could have been sent to the Moon and be there in meteorites. So there's a bit about it, and there's some papers about the possibility of these meteorites from other planets at the lunar poles, and that these could have like diatom shells in it. And I've worked out that if there's organics in them, then nobody seems to have done this calculation for the Moon, but someone has done the calculation for Mars, and they found that RNA could be preserved for I think even billions of years in the very, very cold conditions on Mars, if it's sufficiently below the surface, so it is not affected by the ionizing radiation, which breaks it up eventually completely. And so you have to drill to quite a depth, meter or two, on Mars. And you could do the same on the Earth, on the Moon. It's the same thing. If it's near the surface, it'll be destroyed really relatively quickly, millions of years, billions of years, then there wouldn't be that much left. And but I think Chicxulub impact, there could still be, even fairly close to the surface, there'd be some of the organics still there, a fair amount, but I have to look at paper up against what the numbers are. But when you go back billions of years, then it's pretty much all gone. But so from the Chicxulub impactor, then there could be, it wouldn't need to be that far from the surface, and you'd have the relatively intact life, but the Earth could be quite deep, and maybe the meteorites that have got quite deep beneath it, and there's intact microbes, pretty much intact microbes, or even bits of higher life forms in it. So the Moon is even of astrobiological interest, and there are lots of interesting, intriguing little mysteries, like the lunar swirls on Earth, and the lunar little overturning. There's venting from the lunar surface. There's argon that shows us venting, and then there's one hypothesis, which is a minority view, but it's not junk science, that there could be much more ice on the lunar surface than is thought, that is kind of well below the surface, extending well beyond the polar regions. That's a possibility. So that's something we could investigate. That you drill quite a couple of meters or more below the surface, and you find significant amount of ice well beyond the polar regions. It is an outlier idea, but we have no way to know these things. And the lunar caves, they're huge. They're possibly as many as 100 kilometers long, at least tens of meters wide, and theoretically in the lunar gravity they could be up to kilometers wide, and they'd still be strong, and strong enough to resist the impacts, small impacts into them. And there could be huge caves there, big enough to put a city inside. They have a picture of Philadelphia City, which is a small city, and it doesn't fill the cave across sideways. So we don't know for sure, but there certainly are caves. We could see holes into the surface with these caves, and they continue at least a little bit beyond the entrance. You can see beyond the entrance just a little bit. So explorers can be wandering around exploring these caves, exploring the surface. I think the moon is incredibly interesting, far more interesting than most people realise. And so we have plenty of time, and I explained about the life support and how it's so difficult. And the Mars would be one way. And this is something that, as I talked about, that all the difficulty of returning a sample from Mars, that what Elon Musk seems to be totally unaware of. I don't think many people know this. But if a starship went to Mars, in the forward direction, it's only protected by the Outer Space Treaty, which is very weak. You can drive a coach and horses through it. And it mainly works because there's a whole lot of mutual agreement amongst the different states and the Coast committees, and deciding together that we want to interpret it in a particular way. If some very perverse government wanted to interpret it a different way, then it would be difficult to say that they really held to it. What everyone's deciding is the way to interpret it. But in the backwards direction, as I said, it's very, very strong. And so, if he sent things to the Mars, but he wants them to go shuttle back and forth. He wants it to come back again. So even if it's just setting up for future humans to land there, when it comes back to Earth, it'd be very difficult to make sure that no dust gets into it. That means all the protocols to preserving Earth's biosphere apply, because he could be returning some extremely hazardous microbe to Earth, as far as we know. Hazardous to the biosphere of Earth, hazardous to human beings. He's got no way to know unless he's done a study on Mars and find out what's there. So he can't. They're not going to be legal. He's not going to be permitted to send a starship to Mars. I hope you can understand why. And it's not just silly paper pushing. There's a real reason why he can't do it. He can't send a spacecraft. He won't get permission to launch a spacecraft to go to Mars that can come back again. And so humans, it have to be one way. Mars one is the only way that's legally possible for humans going to Mars. And he doesn't want to do that. So I think so. And then the value of he wants to find out about Mars life as well. I think there's a quote I have here from Elon Musk talking about how it's important to preserve the Mars life if it's there. So I think the only risk with Elon Musk sending a spaceship to Mars is if he personally were to decide that he doesn't think there is life there. And if he didn't pay too much attention to the astrobiologists, he listened to Robert Zubrin and believed Robert Zubrin, but Robert Zubrin thinks there is life there. No, he's not paying attention to Robert Zubrin, just going his own way, and just saying, "Oh, I've had a read," and he's very super confident sometimes, like the way that thinking that his way of rescuing the Thailand cave divers. So I can imagine being super confident, saying, "Oh, people are saying that there could be life there, but I don't believe them," and then just sending life to Mars, sending off to Mars. But he can't do that But I think so. I hope that we can convince these people that it's not like that, and that the Mars that it's possible there is life on Mars. I mean, I don't know if Elon Musk would ever watch this picture, but maybe some of his supporters would. I mean, could astrobiologists couldn't talk to him about it? Could there could be life? It's not paper pushing. There's a reason why we want to make sure that whether there is life on Mars and what kind of life it is and what Earth life can do to it. And so this is the idea of immersive VR. It's a rather fun, techie idea. And it's similar to the orbit of the spacecraft that take these RSL photographs. And as the spacecraft come in, every day, twice a day, every 12 hours, they come in to opposite sides of Mars. And they see and they can get close-up pictures like those RSLs, and then they can do this this telepresence. You have little rovers on those RSLs, and anyway, and so I explained about how the no clumsy spaceships, no collisions between the Mars and Earth biospheres, and we could use in this way robotics could help us to explore space. It's a really exciting mission. It saves all the danger of the landing on Mars, both ways, for humans and for Mars. And so so in order to do that well, we need these sterilized spacecraft. And so the Viking lander was sterilized for 30 hours at 112 degrees C. But modern, like again, Elon Musk using his electric cars, he'll be using these components that they use them to reduce the weight of electric cars, sensors and things that can survive at 300 degrees centigrade. It reduces the mass of the car because you can put them right close to very hot components. And so it's practical to sterilize one rover to far higher standards than for Viking. So there's this study that they did for Venus. Let's bring it up. And he reckoned that this is a much more than 300. It could survive 500 degrees centigrade. And to do that, then he uses his really major revision of how it works. You can't use proper computers for that, but that at least you can only use the very early stage of computing, because the computers that work at 500 degrees centigrade, and they do exist, are they're not at very high level of complexity yet. So at 500 degrees centigrade, you have to use rather simple computers, rather small. No, you can't use your Pentium or whatever, not at 500 degrees centigrade. But at 300 degrees centigrade, you can use much more sophisticated computers. So anyway, he reckons that there's hundreds of millions, and they're developing these computers especially for Venus that can survive at 500 degrees centigrade for a year, for years. And he's saying that he can develop this for hundreds of millions of dollars, and it would take 6 to 10 years. Well, I think we could, and then these so I didn't explain the context there. So the idea is that if you can heat a spacecraft to 300 degrees centigrade, you can sterilize, that's hot enough to completely sterilize amino acids and DNA bases. So you end up with no DNA and no proteins. And so it's far better than just sterilizing of life. So you can do far better than Viking using modern components. Now that's a complete rover that can do it 500 degrees centigrade. But they can do it at 300 degrees centigrade, which don't need to use these sort of extraordinary measures they do there. You could do those quicker and for less cost. And I think we should have a high priority program to produce 100% sterile design, 100% sterile rover that we could send to Mars. It would only need to withstand 300 degrees centigrade. You keep it, or even 250, even 200 would kill all life. 250 destroys, I think, the RNA bases, DNA bases, and I think 300 destroys all the proteins. Is that got it right? 300 destroys the proteins too. 250 destroys all the RNA bases, and 300 destroys proteins. 8 of the 20 amino acids, and not decompose, and the other ones you're not able to completely characterize the gases emitted, but there's not much left there for them either. So 300 degrees centigrade, and you leave it going at that temperature, and maybe you leave it at that temperature all the way to Mars on the journey out there. 6 months at 300 degrees centigrade, because remember these rovers, they're meant to be able to survive as a rover for a year or more, so they can certainly survive the heating all the way out to Mars. Then you're going to have 100% sterile rover. Not only that, you've dealt with the risk of introducing DNA, gene transfer agents, and GNA, and even tiny fragments of DNA or proteins, which might give very early life new ideas. You've got 100% sterile. Now, I'm not saying there's an awful lot of research to be done there, because you, particularly how you get the organics into it, if you've got life experiments, how you deal with like this special spectroscopy and the telescopes and the cameras and things. They have cameras for ovens at high temperatures, so you can certainly do optics at those temperatures. So certainly it's feasible. I don't see any kind of impossibility there that we could have 100% sterile rovers going throughout the solar system. Do that program once, maybe a few $100 million in a few years, and now you've got a design of rover that can travel anywhere without any risk of it being harmed, of harming any native life there. You can have solar panels that can work at those high temperatures, so suddenly you can sterilize the solar panels, or you can use RTGs. They have solar panels that could work on the Venus surface, so you can certainly have solar panels that could work on this. Everything. They've gone through all the components of a Venus rover, and as I say, they can even do it 500 degrees centigrade, and then rather more easily. That requires a little bit more design work, but I think 5 or 6 years they could have it. And then at the 300 degrees C, it's significantly easier than 500. So that's what I think we should do. And I think that there is a significant risk that the Earth that Earth life has got there and is surviving there already. But I don't think it's. I think it's chances are we're lucky, and whatever microbes could survive there, just sitting harmlessly, and that's what the planetary protection officers say. They say most likely that the Earth life. You can't say certainty, but a high probability. Then Earth life hasn't contaminated Mars yet. If it has, you certainly don't want to introduce more life there. I mean, that's like if you've got rabbits that get into Australia, then you say, oh, give it up, we're putting cane toads, frogs, camels, elephants, you like. Certainly there are camels in Australia. It doesn't matter. The rabbits got there, so anything can go there. So that's not how if this Earth life got to Mars, it could coexist with Earth life, like with Mars, like a shadow biosphere. Like the hypothesis of the shadow biosphere of RNA world life. There could be a shadow biosphere to the Earth life that we introduced by accident. But that doesn't mean that you can send humans there, because we'd send massive amount of microbes, both that way and the problem coming back the other way. So there's nothing. So the in situ, the astrobiologists say go and search in situ, and that's what we should do. I mean, if I had, obviously I've got no say in this, but if I had any say, then I would say don't just stop the Mars 2020 rover with its sample thing and the MOXIE. You don't need MOXIE, and particularly you don't need to walk all the way around Mars producing oxygen. Oxygen is an interesting gas. You want to, like they found recently, there's a variation in oxygen on the Mars surface. You want to study it, and they're going to send this rover around Mars that's going to be producing oxygen all the time, because they want to test how you produce oxygen for humans. I don't think we're going to be sending humans to Mars until 2040s. So there's no urgency to send this oxygen generator. I'd take out the oxygen generator, I'd take out the sample return, because we're just not going to be able to return it in time for it to be interesting. It'd be 2040 before you can return the Earth life, and you have to sterilize it. It's not the time to do a sample return until we find out what's there, find out what life's there, and we can return the sample, but return it above geostationary orbit. See, I'm just a nobody, so I can say these extraordinary things. I mean, if I had a tenured position, professor or something, one might hold back and be a little bit cautious. I want to say something that's interesting to get you thinking. I'm not saying this is the answer. So that's what I do. I tear out I should say, go back to the drawing board. We don't need the sample return. Not at this point. We don't need the MOXIE, and instead put in as many life experiments to search on the Mars surface now, right now. And there's so many things that they've produced. That's what we need to send to Mars. I think that's what they should do next year. And I don't think they're going to do that, but just to put my, and I think that's what they should do in the future. And then if we do find life there, then they might discover it. It's possible ExoMars discovers it right away. ExoMars is going to drill, so that means ExoMars. If we go back to that picture up there, ExoMars is going to drill down to there, down to where those biofilms are. So there's a distinct possibility that ExoMars would find, if there's Martian life in those biofilms, I think there's a distinct possibility that ExoMars will find life there. And let's go back up to that picture while I'm talking. There we are, down there, 50 centimeters deep, and 15 centimeters deep. And then it goes down, they think going down to maybe 50 centimeters. So Mars 2020 can't do that. I think it's basically designed by geologists, and they're most interested in the rocks, and they're not really interested in drilling so much as the astrobiologists are. Astrobiologists say drilling is top priority. And ExoMars is going to drill. It's going to be able to drill up to maybe 2 meters. I do hope they have a successful landing. And then they drill down into this layer. And then they might well bring up a sample which might have life in it. I don't know what the chance is. I mean, there might be just very, very small amounts of life in there. But the Viking very well. But Viking, this whole thing could explain why Viking found life if it did. He could have just dug into stuff like that. The Viking, it's very interesting, the Viking results. And if I was sending, and then what I would send, I would do the one thing we'd definitely do is replicate the Venus, the Viking experiment, with a chirality. But also, you would definitely have a suite of different instruments, because the Viking chirality, it doesn't. You could have achiral. Early life could be achiral. There's Joyce's enzyme, or whatever you call it, Joyce's ribozyme, which I think it's called. I can't remember its name, but anyway, this can replicate its mirror image. So if you had life that replicates its mirror image, then you get both sides replicating at once, and it's achiral. So it's not necessarily that chirality is going to prove that it's life, prove that it's achiral. So the idea, I was being technical there. So our life is all one mirror image, and if you take the mirror image of it, then so they have the idea of the Viking experiment was they did an experiment in which they took the dust from the Mars surface, they put it into some organics. It produced carbon dioxide. The organics were labelled with radioactivity. It produced some carbon dioxide or methane. I don't think they knew which it was, which was radioactive. And so, and they don't know if it was life or if it was some very strange chemistry. And they couldn't, because the chemistry was there very strange. They didn't behave quite like life would. However, they discovered a chiral signal, the circadian rhythm. They found the temperature was changing daily, and out of sync with that, significantly out of sync. 2 hours would be far too much to be out of sync for a chemical process, which would only be about 20 minutes. Then you got this circadian rhythm of the amount of carbon dioxide released, or methane, whichever it was. And so I think that they should do that as well. And then the also thing they did with Viking, it was a very strange thing that happened. They found that heating it up to a certain extent stopped the reaction, slowed the reaction down. You'd expect it to increase with most reactions. But with life, then if it was adjusted to low temperatures, then warmer temperatures would slow it down. So that was quite strange. Very unusual chemistry does that. Another thing they found with Viking was that if you kept it in darkness for a couple of months, which it did by accident, then nothing evolved from it. And that's very difficult to understand in terms of chemistry. Why would darkness inactivate the chemical compound that was producing the carbon dioxide or the methane? So I think you should do all of those. I think we definitely need to reproduce the Viking experiments and see if you get a circadian rhythm, see if you can reproduce that month of darkness thing. Anyway, so I think we should do two things. We should do that to the equatorial region, reproduce Venus and all the other experiments we've done and drill. And then in the polar region, we should again drill. Drill is very important. If you drill down and you find early life, even 100 million years, there were times when the surface was more habitable and there was more liquid flowing, and you should drill down. This was the icebreaker. Chris McKay is very keen on this, and I think it's very important that we have to be able to drill. And if you go to the icy regions, then even if there's no life there at the present, maybe there was back then. Maybe there's life that comes up to the surface from time to time, spreads out, and the remains are still there but dormant, and they will come back to life again if there's more life, if there's more liquid again. So we need to drill down there. But I think we should go to the other expedition we should do is go to the Phoenix lander. So one of the big difficulties in motivating sending life instruments to Mars is they mightn't detect anything. If there's no life there, then they wouldn't detect anything. And it's quite a hard sell to say, we're going to send this instrument to Mars, but it mightn't detect anything. And it's hard to explain to the public that not detecting anything is a major discovery. A null result is a major discovery. So in order to have a discovery that is guaranteed, then in order to, so you don't just have a null result if there's no life there, then I think we should have a mission that goes and looks. And so for the Curiosity, if you can get a sufficiently sterilized thing to send it to, and I think for Curiosity follow-up, it wouldn't be too bad an idea to send it to where Curiosity went and go and study Curiosity. But a better one might be, for this plant protection one, is to go to the Phoenix, because it got crushed by the dry ice, and there were also these little droplets there. Then look and see if any Earth life got into there. You would certainly find it on the spacecraft itself. So you could send this mission and land maybe a kilometre away and drill there, 10 kilometres away, 2 kilometres away, whatever you think is safe. Drill there and look what what it's like there, and then go closer and keep looking, spot samples. And if you got an increase, and especially if you went very, very close, you wouldn't expect it to spread very far, except possibly in the dust, but I mean, it's not too likely. Then then you hopefully you just find a little patch near Phoenix, and hopefully just on it. Maybe worst case you find a little patch that's actually colonized. So then you'd have to think, what do we do now? Can we sterilize it? So I think those are the missions we could do. Now I'm just going to do. This is going on. Quiz me. I didn't mean to speak so long. Chaotropic agents. That was about how. I think I talked about those. Okay, let's do the control end if it lets me. Yeah. I would just put this up anyway. I could do it as two separate videos. So Robert Zubrin said, so we can go for many ways, but we go in a way that respects science and values extraterrestrial life wherever we find it. So we're going for space mining and discovery, tourism, for any reasons. Let's do it in a way that respects science. Yes, I know. I'll add this video to this article, and then if someone wants to read through it and then they want to watch the video as well, then it'd be sort of going into some more details about some things. So, as Arthur C. Clarke put it eloquently in that story, this world around them was no longer the same. Venus was no longer dead. It had joined Earth and Mars, for life called to life across gulfs of space. Everything that grew or moved upon the face of any planet was a portent, a promise, that man was not alone in this universe of blazing suns and swirling nebulae. If as yet he had found no companions with whom he could speak, that was only to be expected, for the light years and the ages still stretched before him, waiting to be explored. Meanwhile he must guard and cherish the life he found, whether it be upon Earth or Mars or Venus. And that is the attitude I think we should have, guard and cherish the life we found, whether it be on Earth or Mars or Venus, we would add, or on Enceladus or on Europa or Ceres, or even Pluto. And it might be that every single one has independently originated life. So I think we need to guard and cherish it. And so Robert Zubrin, and so I want to talk, I'll just briefly mention my pale blue dot as well, but just to end this, I think. Anyway, so Robert Zubrin argued we have not just the right but the obligation to establish ourselves on Mars. We are the creatures with all of our flaws that the Earth's biosphere has evolved to allow itself to reach out and establish itself on additional worlds. And he says we'll take a nearly dead world, create a new, fully living world, so there'll be new species of birds and fish and plants, and it will be magnificent. And no one will be able to look on it and not feel proud to be human. I think we should do that on the moon. The caves on the moon could be filled with atmosphere, and then that can be as magnificent. It could be a magnificent and there could be new species of birds and fish and plants that can fly around on the moon. Little creatures that can fly that never knew they could fly. They would evolve the ability to fly on the moon, which they never had on Earth. And that's possible. And then we can have the space, because I said the lunar caves, they could be 10 kilometers across or so. And there's the lunar vault has lunar poles, and atmosphere isn't have much mass either. And and you could have lunar railways and moving things around. And then if you have the space habitats in the asteroid belt, then with one calculation, there's enough there for radiation shielding, for habitats, if you were to dismantle all the asteroids, for habitats with 1,000 times the land area of the Earth. You should have equivalent of 1,000 planets of land area, just in habitats made from the asteroids in the solar system. And Asimov used to call it planetary chauvinism. I think he thinks he might have got it from Sagan, I think he said. But he's not sure where he got it from. But that's the idea, that because we evolved on a planet, that our future descendants should live on planets. I don't think that's true. And might well be. And the designs for spacecraft that could habitats with revolving solely for artificial gravity all the way out to beyond Pluto with large thin film mirrors to collect the sunlight even without fusion power. So that could be magnificent. And what I think is that, and I say that there, we provide a way of getting space, but if Earth evolved us to reach out and establish ourselves on other worlds, it also evolves with the intelligence, foresight, and deep scientific and technical understanding to guide that exploration. We are the Earth's biosphere's guiding intelligences in space, and that may be one of our main rules. Not only that, but and one of the reasons for going to space may be to protect Earth from hazards such as asteroids, or to find resources for use on Earth, or increase our understanding of ourselves and of science, biology and the universe, and indeed as a place for adventure and recreation. And we've already found many benefits for Earth's biosphere already through our satellites in Earth orbit. So, and I say it's not automatic that everything that human beings find inspiring is going to be work and be harmless. The people were it was a deliberate introduction of the European rabbit to Australia for sport. So for them, those days, they found it inspiring, but it led to many problems. So, yes, we can set it up on the different habitats, but not yet on the Mars. Let's find out what's there. We are biosphere's noosphere, the intelligence that it needs to avoid catastrophes. And we can use that to protect Earth, to get through the climate change issues, which we can certainly do, and to protect us from asteroids. And if we need to go into space, with 100% sterile rovers, in my view. And then I'm just going to bring up, I hadn't meant to do this, but I'm just going to mention it slightly. I think actually it's the other way around. I just happen to, this is the way that I know to get to this thing. So this, I think I'm going to pop that up as a preprint too. I did that for the pale blue dot challenge. I haven't done it yet as a preprint. I might put it up before I do the, so our pale blue dot are the other homes for us in our galaxy. So this is about, I think that we should look at things the other way around. Do I have a picture of the pale blue dot there? Don't I? Take a while to come up. So anyway, taking a while to come up. Maybe it's further down the page. Oh, there we are. So that's Earth, as seen from Voyager. This isn't an actual artifact of the photograph, and that, but it talked about the pale blue dot hanging in a sunbeam. And that's the Earth, as seen from space. Tiny blue dot, pale blue dot. Anyway, as we go into space, I actually think that it's not really our main challenge. I don't think our main challenge is, now that we've got space technology, I think it's inevitable that we're going to have space colonies. So maybe having a little slower than the colonization enthusiasts thought. But I can't see a future without it myself. And I think it's the other way around. Watch out, galaxy. So the question is, are we going to protect the galaxy as we go out? So I'm not going to talk that whole thing. So I think that if we go out and we do it in this way of respect for, if we must guard and cherish the life we found, whether it be on Earth or Mars or Venus, then I think we have the right attitude to then expand into the galaxy. And I think we're going to be extremely cautious about setting up a colony around another star. Because you need to think, I've talked about that in another talk, but we need to be supremely confident in our descendants before we set our descendants off into the entire galaxy, where they're going to be so far away that we have no communication with them, and where they could be doing something for 1,000 light years, through 1,000 light years away. Then our descendants, assuming we stay on the Earth, or maybe your descendants, but either way, it wouldn't matter which you are either. But say on Earth we just keep we are very cautious and careful. But say that 1,000 light years away, they just forget about all that, and maybe they'd build a self-replicating thing that would be quite harmful, so it could actually be that our descendants, we have all these things about alien monsters invading Earth. I think that a sensible extraterrestrial would be very cautious about letting its descendants just go off and replicate throughout the galaxy without having supreme confidence, sort of knowing, having very certain that your great and a dozen more times grandchildren are going to have exactly the same ethics as you do, and or at least similar enough so that you know they're going to treat the galaxy with respect. So just as we guard and cherish the life we found on Earth or Mars or Venus, I think we need to be sure that our descendants, that we are producing a civilization in which all our descendants are also going to, and that's a tall order. It's not easy. And that might be why we don't see many extraterrestrials around. I don't think there's any chance of ourselves going extinct myself. We're extremely robust. As I said at the end of this, so now I've really got to stop soon, but I'll just say this bit at the end. A rather nice quote from the end of this article. Right up at the top of the bibliography. And it says, the anthropologist Mary Russell writes musingly about her attempt to characterize what's. She said, human beings are a dangerous invasive weed species that's invented central heating, air conditioning and food that can be stored for up to 10 years, so not even a direct hit by an asteroid would likely make us extinct. And I think that's the situation. I think it's even more than that, you know. I don't think we can go extinct myself. I really think it would be an extraordinary thing that would turn us and make us extinct. And so we could be especially devastating for non-technological civilizations we encounter, and also for. And so I think sensible ETs look at that, and then sensible ETs I think they look at that possibly and find a way to become a flower of the galaxy rather than a radical weed. And if they can't, they stay at home until they can. And I think less sensible ETs, and they destroy their own spaceflight capabilities in uncontrolled wars. So that is another concern. It's not going to happen in the near future, but I'm thinking about a few centuries in the future. If we set up colonies in space, like what the space colonization enthusiasts want to do, we need to think, we set up the colonies where there's going to be some adversarial interaction between them and us. So you need to be careful. And I think that going into space with this attitude that you guard and cherish the life you found, I think that this is an attitude that we need. And I mean guard and cherish the Earth. Think how wonderful the Earth is. And just keep that very clear in our mind. It may well happen naturally as we go into space. It might be this that, like they say about people looking at space, the Earth from space, and it looks so fragile. And that may change our attitude as well. It might be that space explorers naturally have a tendency to kind of cherish life wherever it is. Where they look how flimsy, look how thin, how fragile. It isn't really flimsy, but how fragile the Earth looks. And that kind of feeling of how, and also the way we're working together with the Earth's biosphere and so on. So, much though I would really love to have colonies in space, as a child I would think that was the most wonderful thing. But I do think we also need care, and I think not to be too frustrated if it's a little bit slow. That it is really important that we do this right. I think that's the one message I want to end with. We need to do this right. We need to be the noosphere was Théa de Chardin's word for it. We need to be our biosphere's intelligence. We are the intelligence it can use to go into space, but also the intelligence it can use to avoid catastrophes. Oh yes, and then so I was just going to say, so I hardly said very much about this actual report for this last little bit, for a fair bit, but I should quickly go back to that. So they said that the supporting finding, various studies suggest that survival and application of terrestrial water are unlikely on the Martian surface, which supports classification of much of the Martian surface as Category 2, and made the recommendation that much of it would be Category 2 instead of 4. And so I hope you can understand that I've said enough to say that the reservations with this suggestion, we can't do that until we've done more research into the Mars. We need to look and see what's there. And I think, especially looking at these biofilm and so on, I think it's extremely unlikely that further study will come up with the conclusion that anything from Mars is Category 2, and especially when you look at the global dust storm, then any life anywhere could spread anywhere. So I hope I've said enough for that. And then there's the bit that in this report they say that they said it would be impractical to require launch providers or satellite hosts to ensure that their payloads from Mars are free of biological contamination. They said there should be a system of sanctions that space missions are penalised for contaminating Mars. And what good is that? What's the good penalising someone who has contaminated Mars? It doesn't make sense. It literally does not make sense. If your aim is to protect Mars, that is not protecting Mars. All that will do is punishing the people who have made Mars, who have contaminated Mars. And punishment is no good after it's happened. And they gave the example of the Beresheet lunar lander, which had tardigrades on board, and it was not declared, and it didn't follow the planetary protection requirement. Now, for the Moon, all you had to do, it was just a formality really. They just had to say, we've got tardigrades on board, and it could crash, or it's going to land on this part of the moon. They just need to have literally written that down and put it up on a website or something. And they didn't do that. They just didn't tell anyone. And I don't think there was any willful. I would imagine they just didn't even think of it. A lot of people are not that clued up on planetary protection. But so there's no risk of forward contamination on the moon, but there's a requirement to declare those things in case someone finds tardigrades on the moon and they wonder where they got there, how they got there. So that's the reason. So the text of this report, it says supported finding that it's impractical, and so supported recommendation, breaches of planet protection reporting other requirements should be handled via sanctions that hold the root perpetrator accountable, rather than increasing the verification and regulatory burden on all actors. In other words, they're saying that the United States government and SpaceX, if they launch someone else's thing to Mars, and the people who provide the launch facilities, they do not need to look at the payloads to see what's in there. And Elon Musk literally could send a spacecraft to Mars full of lichen and cyanobacteria, and he just thinks it's a good idea, and he doesn't tell anyone because he doesn't want to get into trouble. And then when he gets there, he just announces to the world, I have seeded life on Mars. Hooray, look what I've done. And then they say, you've got, we're going to penalise you. You've got a fine of $500 million. I mean, that is not going to work for protecting Mars. So, no, I do hope that they do not follow this recommendation. Now, I mean, luckily, the US is not the only thing. Coast Guard is international, and they can't change the planetary protection recommendation, at least the international ones. The US could go it alone, but I hope the US do not go it alone. They would still be within the Outer Space Treaty, but just say we have a different interpretation from the international interpretation. I can't see the Coast Guard agreeing with this, because they are people with their heads screwed on. They're astrobiologists. They're not going to agree with this. So I can only see the maybe NASA would try to go it alone. I hope they don't. I don't think they will. I think hopefully this is just a little thing that's not going to come to anything. And so, oh yes, and then last of all, in case anyone's watching this who's connected with SpaceX, who's gone all the way towards this, got to this end, there's lots of benefits to humanity from astrobiology. If you bring that up, and I'll bring up the new biological materials and new dimensions to biology. So that is like all our biology so far has been unidirectional. It's like you've got to draw it on a graph, but it's not even a graph with two axes. It's only got one axis, and everything is along that axis. And compared with that, then another form of life is like a second dimension. A different language, different, even if it's just very slightly different, even if the table, the lookup table for making proteins from RNA is different. That is still not, we don't have that. So we have a new dimension to biology. We have benefits to humanity from astrobiology. I think that's the whole second title. We have new biological materials. So we look at all the biological materials we have on Earth. We have wood, we have yeast making your beer and things. And goodness knows what the microbes on Mars will do. And there might be higher life forms there. And there could be all sorts of materials, biological materials that we don't have. And then there's a genetic firewall. So this depends on what we find there. So I was talking all along about how it could be hazardous. It could also be beneficial. So we don't know that Mars life is hazardous. I have a whole section in my online preprint paper about how it's possible that colliding biospheres is actually beneficial. And this actually happens with invasive species sometimes on Earth. And so it is possible there's stuff we find on Mars. So obviously I talk, because I'm talking about planet protection, about the worst case. There's also the best case in terms of the collision. It is possible that what comes into Earth actually helps and supports the Earth biospheres in one way or another, but you do it with extreme caution before you're introducing something like that. But there are cases of life that comes from one part of one continent to another that actually benefits and the local ecosystem and makes it grow better and improves the conditions for local life. I have some examples on my paper, preprint. And then there's the possibility of a genetic firewall, which that is to do with maybe it just can't survive without some kind of special ingredients and you can have a kind of firewall. Anyway, I can't remember that section very well. And then, but if I go to that one, I can actually remember the firewall, and let's look it up. It could be, yes, that it has no resistance to Earth life, and it can't survive anywhere. If we had life like that, yes, life that has no resistance, it can't survive, or else it could be, for instance, that it only flourishes at extremely low temperatures and just self-destructs. Then it could make it invisible to natural biological systems, and that could also be used to engineer a kind of genetic firewall. And they're talking about artificially modified life, which then replicates and used to generate exotic chemicals with no risk at all to escape into the wild or hybridize with Earth life. And they give various specifications of what this life must be like. And at the moment we're trying to make this kind of XNA-based life by modifying Earth life. That's a tall order. But maybe we find a form of exobiology is naturally orthogonal and also is not able to survive on Earth. It's not able to, and then we could use it, for instance, for manufacturing implants is one example that wouldn't be rejected because Earth life doesn't recognize that it's a threat. So that is a possible way in which it could actually be beneficial. And in other ways, and then there's the possibility of enzymes from extremophiles. And yeah, so that's in my occasional touch. I think that is also in my preprint. Though it's possible there's some things in here that I just haven't put into my preprint. So it's possible that it isn't. And then, so these enzymes, I got this in an article from Charles Cockle, and he just says that has found some remarkably prosaic applications like the powder in your washing machine at high temperatures. Also powders that work in washing machines at low temperatures. And I looked into it, and these enzymes are now used, and enzymes from extremophiles are used in the food industry. There's a $1 billion industry of enzymes for detergents. It's used for wood pulp and paper processing, textile, cement, cosmetic industries, and this is all based on extremophiles, which are extraterrestrial microbes that only survive, that like it best in very hot conditions or sometimes in very cold conditions. So those are heat adapted ones, also cold adapted ones, extremely useful. So, and these are used for instance for washing. If you've got limited, yes, the detergent enzymes, if it's over 1 billion, and they may get inactivated as they get hotter, which is useful. For instance, for meat tenderization, then you heat them up and they inactivate. And then they're also used for cold water detergents, and they reduce energy consumption, and producing lactose-free foodstuffs, and food processing for clarification, reduce viscosity and extract natural oils, bread making. It lets the dough improve at low temperatures, textile research, cement and cosmetic industries. So these are just from extremophiles, and then we could easily be as many applications as that and more from extraterrestrial life based on a different biochemistry. And remember, there is a possibility that this life just goes extinct even before we find it. So you can have purely commercial reason for being very careful about what you do with extraterrestrial life. So this might be a point at which to finish. So as I've talked about, I think I've talked about just about everything, and so you've got my touch. I just briefly mentioned my books, and then I'll finish. So you've got touch bars. As far as I know, this is only the second book on planetary protection. It's over 1,000 pages. and even so, it only touches on many of the things it covers. It's a vast, vast subject. And most books for the general public, then there's only a chapter or so on planetary protection. There's one other book called When Biospheres Collide, which is the history of planetary protection. And this is focused mainly on NASA. So that's the only other one I know of for the general public. This is available to read for free online. You can also get it on Kindle, where you have to pay a small amount. And then I have my Case for Moon First, which goes into, and this goes all the things like Hoyt's Lunar Tether again. It's available for free, and then you can buy it. And then there's my Moon First, and it was originally done at the time of President Obama. I haven't updated it to President Trump. So anyway, it includes the section Astronaut Gardener on the Moon. I haven't actually updated it for a couple of years now, whereas this is quite a bit updated, and this is updated sort of last year and some bits even now. And I will be doing some more. I meant to do a lot of work more on that, and I've had other stuff come up, but there's some more to be done there. And then I have this paper that I'm working on, potential severe effects for biosphere collision and planetary protection implications. And this is not peer reviewed, but I'm very interested in feedback, and I'm planning to send some parts of it to an astrobiology journal at some point. I don't know when or what. And then I have this pale blue dot. I haven't uploaded it yet, but I just had this idea today that I might as well do that as a preprint too. And I'll put it up in the same server. It's the same thing, it's got lots of sites and stuff. I did it for the 9 Dots Prize essay. Someone else, very well deserving, got it. But and it was only specifically for that year that I had a chance of it winning. So I'm going to put it up on as a preprint, I think. And then I might expand that into a book, but that's just for the distant future. And there's a whole section about that in OK to Touch Mars. And at this point, I think then that's time to stop. And I hope you enjoyed it. I hope there were, I've moved myself over there, so you haven't seen me for a little while. Yes, so hopefully this has given you some interesting stuff to think about. And I'm going to stop here. And I'll probably attach this to for someone who's kind of if anyone is very keen, and maybe you can dip in and there'll be bits that you're interested in. I don't think I'll split it up. What I'll probably do is have a kind of index to what I talked about, so if you're interested in particular things. And so yes, do look in the video description and do subscribe. Well, everyone says that if you're interested in some of my things, but I talk about loads of different things on this. I will upload this in my Mars and space colonization thing, so it will only be a Mars and space colonization related.